Hello, this is Jeffrey Fox here, and I'm presenting a tutorial on the package Plotviz built at Indiana University and available online, as we'll give at the end. And it's designed to plot sets of points. So we learned in the X-Informatics and Big Data MOOC that large high-dimensional data was essentially very pervasive, science, business, web, and it's pretty hard to understand data that's in a high-dimensional space or data like that in recommender systems where they have vectors, but those vectors don't even have all their components defined because you only have rankings of a few items from a few users. However, in essentially all of that data, you can define a distance, and you can use that to do dimensional reduction and represent the data in 2 or 3 dimensions. And this final 2 or 3 dimensional data is what Plotviz is designed to support. The methods mentioned here: PCA, principal component analysis; MDS, multidimensional scaling; GTM, generative topographic mapping; SOM, self-organizing maps. These are all ways of doing dimension reduction. Dimension reduction is not part of this talk. It is about this talk is about Plotviz, which is a package to visualize the results of dimension reduction. And so it produces the 3D scatter plots, lots of points defined in 3 dimensions. And you can browse that 3-dimensional structure to try to understand the overall structure. So see clusters. Of course, there are clustering programs which are separate, but you can then see whether those clustering programs have produced reasonable answers. Because human beings' eye is extremely powerful, and one needs to bring it to bear to make the best analysis of any data set. So this is a result of Plotviz showing a Cheminformatics data set with each color representing a different compound, and then you're just rotating it in Plotviz to look at the different structure as it goes around. Note these points look as though they lie on the surface of a sphere. That's a feature of dimension reduction. If you take points in high dimensions, because distances tend to be large in high dimensions and tend to be at the maximum value, when you reduce it to 3 dimensions, you tend to get the points at the surfaces of the sphere. That again is nothing to do with Plotviz. Plotviz is accurately representing the points that it was given. Here's this same plot in a somewhat just larger format, and we will go through how to manipulate these plots and give you further examples. So this slide here gives you this approach in more detail, how some of these dimensional reduction methods like GTM and PCA are aimed at vector spaces. Others are aimed at spaces where we only have distances, such as that recommender system spaces. Genomics also has such spaces. That can actually go to any dimension. The advantage of 2 or especially 3 dimensions is that it's relatively easy to visualize the final result. So here's our first example in a little detail. It's a very simple plot with 8 points. These 8 points are just at the corners of a cube in 3 dimensions. They've been divided into 2 clusters arbitrarily. The points of z equals 0 have 4 points. The points of z equals 1 have 1 point. And we give them the label P1 through P8. And so you can use Plotviz to make a picture sort of like this. And this example is available on the website, and that's a part of a resource for this tutorial. We will actually go through this example in a little time. I want to put the actual use of Plotviz in groups so we don't keep switching back and forth between the PowerPoint and the program. Here's a more complicated example, 10,000 points. This is proteomics data where the only thing you have is the distance between each pairs of the points, so you have 10,000 squared distances. And after some subtle activities, which are not the purpose of this talk, you can get this type of representation here. And the colors are actually the colors of the clusters defined by the experimentalists. And you can see some of these clusters are actually quite closely clumped on the actual Plotviz plot. And Plotviz lists all the clusters. Here's cluster 403 with 30 points in it. 1701 just has 1 point, and so on. So that's a more complex example of using Plotviz, where you're trying to actually study whether the clustering, which has been done with biological input, actually somehow is seen in the geometrical representation of the data. So if you use Plotviz, you will find that it has most things of use can be done with the mouse. With the mouse, you can click and drag, and that will rotate the points. If you can zoom in and zoom out with the mouse wheel, or you can change the actual center of the plot, which means you physically move the whole plot, either using Shift mouse, or you can use pick a center to choose a new center. We'll illustrate that. Zoom in and zoom out can also be done with the mouse. For some plots, which we won't illustrate here, for instance, the data is only in 2 dimensions. You don't want 3D because you don't want to rotate into a 3rd dimension. So you can force that by just pressing 2 when Plotviz is operational. And thereafter, all operations will be performed in 2 dimensions. You can go from 2 to 3 by just pressing 3. But the default for Plotviz is 3 dimensions. And that's the usual case, because usually you have actually higher dimensions reduced to 3 dimensions by one of these dimension reduction methodologies.